Mitosis is a part of the cell cycle.
The cell cycle is a cell's basic life. This happens to all cells.
But mitosis only happens to somatic cells. These are like your body cells.
Somatic cells are all cells except sex cells. Sex cells go through a similar process called meiosis.
But we're here to talk about mitosis, so let's jump in. Mitosis is a part of the cell theory, and it says that all cells come from other cells.
We have mitosis to grow and repair. Mitosis in the simplest form is one cell splitting into another identical cell.
Sort of like cloning. Now, before we can have mitosis, we have interphase,
which is a part of the cell cycle.
Interphase is the basic life of a cell, but it's not a stage of mitosis.
Interphase is not a stage of mitosis because there's no dividing, even though without it, we would not have mitosis.
A cell will spend 80% of its life in the interphase form. It measures from the beginning of its life to before the splitting of mitosis. Interphase is split into stages just like mitosis.
G1, S, and G2.
G1 stands for growing stage 1. The cell is preparing so all the organelles double to make sure there's enough for both cells.
S stands for synthesis, which is the doubling of the DNA. It's still connected, so it's still considered one strand of DNA.
G2 is growing stage 2, which is the cell getting ready to face mitosis, so the cell membrane is growing.
Okay, so you can clearly see that we are in interphase, G1, by it being one complete cell with the nucleus and doubled organelles. You can clearly see the centrioles over here in the shape of T's, some mitochondria, some vacuoles, just all the organelles doubling. S phase is fairly easy to identify with you'll probably have two cells, both with nucleuses, one with one strand of DNA, then in the other more of that DNA. It's basically more of that DNA just doubled in that same nucleus. G2 is the easiest to recognize because it's just one cell with its cell membrane enlarging. The cell membrane will enlarge, so when cytokinesis happens, everything will be equal and will happen smoothly. That brings us to prophase. Prophase is the first stage of mitosis.
So what happens in prophase?
Well, the chromatid, which is one half of the doubled DNA from S phase, doubles again, along with the centrioles, which they control the spindle fibers. Two chromatids form a chromosome, which is attached at the center with a centromere. The nucleus is currently disappearing from the cell. This is the end of prophase. You can clearly see the nucleus is disappearing while the chromatids are leaving the nucleus. The centrioles are at the poles and the nuclear membrane is clearly intact. Time to get into metaphase.
Metaphase means after phase, which is when the chromosomes start to move to the middle of the cell and the spindle fibers begin to grow. Motor proteins are going to push and pull the chromosomes into the middle of the cell. Dynein is one of those proteins and it's close to the centrioles, which are at the poles. The spindle fibers will now attach to the centromeres of the chromosomes. The nucleus is completely gone now. To remember metaphase, think metaphase is similar to middle.
This is what a cell will look like when it's in metaphase. The chromosomes will clearly be lined up in the middle with the spindle fibers coming out of the centrioles, which are at the poles. The spindle fibers will be attached to the centromere of the chromosomes, which is right at the middle of them.
Furthermore, we get into anaphase.
Now the chromosomes are aligned and ready to move. The spindle fibers will start to retreat back to the poles of the cell, splitting each chromosome into two chromatids in the process. The spindle fibers will pull each chromatid towards the centrioles, which are at the poles of the cell. There is still no nucleus, and when thinking of anaphase, think away. This is what a cell looks like when it's in anaphase. The spindle fibers will start to retreat back to the centrioles, which are still at the poles. The spindle fibers will pull the chromosomes apart, and they will form chromatids, pulling them to the poles of the cell. Let's split into telophase.
Telophase is the last phase where the cell will actually break into two new cells. First, the nuclear membrane takes an oval shape, and the daughter chromatids and centrioles are at the poles, with the nucleus reforming around them. The membrane will now start to form in a peanut shape, as we're getting closer to cytokinesis.
This is telophase. As you can see, the nucleus is reforming, and a centriole is in each part of the cell, along with other organelles. The chromatids are lined up where the nucleus is reforming, and the nuclear membrane is starting to split. As you can see, the cleavage furrow, which is this part right here, is forming. Cytokinesis is the actual splitting of the cytoplasm. It happens differently in plant and animal cells. In animal cells, cytokinesis is the cytoplasm pinching off and the cell breaking. In plant cells, the cell wall is too thick to be pinched, so the wall forms in between the cells and breaks off the two cells. Finally, we have cytokinesis, which is when the cell will actually split into two. In animal cells, the cytoplasm will pinch and split off, and then the cell membrane will reform. The 46 chromosomes will be in the nucleus, along with one half of the doubled organelles in each cell. In plant cells, the cell wall will form in the middle and push the two cells apart, along with the doubled organelle in each cell. A checkpoint is where the cell will check to make sure the cell did something correctly. When thinking of a checkpoint, it's just basically double checking. We have 3 checkpoints: G1, G2, and metaphase. In the G1 checkpoint, the cell will check to make sure each organelle was doubled, so the cell will be equal when it splits. In the G2 checkpoint, the cell will make sure that the DNA doubled correctly in S phase, so we will not have any different cells. So they will all be identical. In the metaphase checkpoint, the cell will check that the spindle fibers are connected correctly to the centromeres of the chromosomes. so they will split equally in anaphase. They do this because if the chromosomes don't split equally, we can have genetic disorders such as Down syndrome. The metaphase checkpoint is also referred to as the mitosis checkpoint or the in checkpoint. Interphase is the basic life of a cell with stages G1, S, and G2. G1, the organelles are doubling. In S, the DNA is doubling. In G2, the cell membrane is growing so it can hold all the organelles and DNA, and is getting ready. Mitosis.
Prophase is the first stage of mitosis, and in it the nucleus disappears.
Metaphase is the second stage of mitosis, where the chromosomes line up in the middle. The spindle fibers also grow in this stage and will attach to the centromeres of the chromosomes. This will later play a role in anaphase.
Anaphase is the third stage of mitosis. The chromosomes split into chromatids during this stage.
The way they split is the spindle fibers will retreat back to the centrioles, which are still at the poles of the cell. Remember, the spindle fibers were attached to the centromere of the cell, so they will pour the chromatomes into chromatids. Telophase is the fourth and final stage of mitosis, where the cells actually split into two, including cytokinesis. Cytokinesis is the process of the cells splitting into two. Cytokinesis happens different in plant and animal cells. In animal cells, cytokinesis happens when the cytoplasm will pinch off and the cell membrane will reform. In plant cells, the cell wall is too thick to be pinched, so it will just form in the middle of the cells and push them apart. Checkpoints happen during mitosis, and there are three: G1, S, and N. G1 checks the organelles to make sure they're doubled correctly. G2 checks the DNA from S phase, and M, also known as mitosis or metaphase, checks is the spindle fibers are connected correctly, so the chromosomes will split evenly. Now it's time for the quiz.
In interphase, there are three stages. In which stage does the DNA double?
Number 2. What cells go through mitosis?
Number 3. In metaphase, where do the spindle fibers attach?
Number 4. In anaphase, what pulls the chromosomes apart?
Number 5. In telophase, why does it make a peanut shape?
The correct answers are 1, B, 2, A, 3, B, 4, C, and 5, C.